Right, so we are now at lesson 6, unit 5. So lesson 6 looks at predicting inheritance for two genes, and to be very specific, two unlinked genes. So up till now, you've always predicted inheritance for only one gene, but there could be one or two or three or four multiple alleles in a gene. Now, this time we're going to predict inheritance for two genes at the same time. So things are going to be looking slightly more complicated. However, just want you guys to be aware that for studying inheritance for two genes and how they're being inherited, we're going to simplify things a little bit and only do it under the complete dominance inheritance pattern. All right. So first up, what do linked and unlinked genes mean? So if you have a look at the diagram right here, the top one shows you unlinked genes. Unlinked genes means that the genes, for example, gene A and gene B, they actually sit on two different homologous pair of chromosomes. So this is a pair of chromosome. This is another pair of chromosome. Remember, in humans, we always have chromosomes in pairs. And the pair of chromosomes will always have the same gene in the same location. Now, if these genes are sitting on different pairs of chromosomes, when they are being inherited, they're actually inherited independent of each other. So the inheritance of the alleles in this gene, gene A, is not going to be affected by the inheritance of alleles in gene B because they sit on different chromosomes. And when they get separated and put into the sperms and the eggs, which are called the gametes, they're being sorted independently of each other. However, if you have something called a linked gene, that means your gene A and gene B actually sit on the same chromosome very close to each other. So the alleles are actually associated with each other. And when you produce the gametes, they are going to be heavily linked to each other. And a certain allele could be associated with another allele in a different gene. And they always seem to be inherited together. So they're not independent of each other when they get inherited. So this is what linked and unlinked gene means. Now let's look at the tools that we use for predicting inheritance. The one on the left-hand side you'll be very familiar with. It's called a Punnett square. However, the one on the right-hand side is called a dihybrid cross. And this one looks a little bit more complicated. Essentially, they work the same. A dihybrid cross is just a complicated version of Punnett square. It just has more squares in it. Now, they actually work by the same principle and has three major steps. And this, let's just look at how Punnett square works. So you guys all know how to do it, but behind the scene, these are the three major steps. We usually identify the parent's genotype, and we kind of visualize how they produce gametes using those genotypes. So let's say, for example, this is a cell, this is another cell. Inside the cell, you have the gene A sitting on a pair of chromosomes. When you produce sperms and eggs, your gametes, you separate them. You have one sperm inheriting one of the allele and the other sperm inheriting the other allele. So 50% of the sperm is going to get this one. The other 50% of the sperm is going to get the little a allele. So that is your step number 2. Then your step 3 is what is being represented here in the grids, which is the fertilization of the sperms and the eggs. So if this sperm fertilizes this egg, you get this result right here. If this one fertilizes this one, you get this one, and so on. So in the square is basically the different combination of sperms and eggs and what is being generated from it, the phenotype and the genotype being generated. So when we look at a dihybrid cross, they work exactly the same way. You also have the genotype of the parents. You have the alleles that could be in the eggs and the sperms, represented here. And then when you fertilize the eggs and the sperms, what do you get? So same principle, same idea, same steps. Let me try and help you visualize how you may get these combinations in the eggs and the sperms. So let's say we have our parents, so the mother and the father. Remember, we are looking at two genes that are not linked. So they're unlinked genes, meaning that they sit on a different pair of chromosome. So this is your pair number one. So your first pair, and then you have your second pair. And A sits on the first one, B sits in the second one. When you produce gametes and when you try and split the cell in half, what happens is that each egg and each sperm will get one of the pair. So we need to work out what kind of combinations we can get in the eggs. Let's say the first combination you're going to get is I may have this one and I might inherit this one in my first one. So that is going to give me a big A and a big B. The second one, I could get the first one, but then this time I can get the second one of the second pair. So that's going to give me big A, little b. Now the third one, I may get this one and this one. So that's a different combination. Fourth one, I may get the second one of the first pair and the second one of the second pair. And that means I'm going to get something like this. And there I have all the possible combinations of alleles in my eggs. There won't be any other combinations, all right? That's all you're going to get. In the same way, we can work this out for the father, what they might have in the sperms. So since the mother and the father have exactly the same genotype, if you follow the same process, you'll actually see that they would be same combinations. Now, depending on the scenario, sometimes the genotype will be different. The combinations could be different, all right? But in our case, the combinations will be the same because they have the same genotype. Now, after you've gotten the eggs and the sperms and the alleles in them, you can start fertilizing the eggs and the sperm together. So let's say our first one, if you put these two together, then you get two big A's and two big B's. Now, when you write it, you put the A's and the B's together. So we would not write things like A, B, but we'll write it AA and BB, all right? Make sure the alleles of the same gene are being put together and alleles in the second gene is being put together. Now, that's your first one. When we look at your second one, we get something like this. The A, sorry, let's just keep going with the first one here, and then we'll fertilize the second one together. So in this case, if you get the big A, big B, and the big A, little b, you're going to have A, B, and B. Now, this essentially is how it works. But to make things super clear and very organized, we won't use a lot of lines and arrows to do that. So we just stick to our Punnett square, or you can call it the hybrid cross. The hybrid cross, So this one right here, the step that we do is basically the step, all right, the eggs and the sperm. And then this step that we are doing is basically here at the bottom. So what you would do is after you figure out all the combinations of the eggs and the sperm, you would write them on these two rows and columns, and then you'll just combine them here at the bottom. So big A, big B, big A, big B, big A. Whoops, I made the same mistakes. There we go. So what you would do is you would just continue and fill up the entire thing. So I would like you to just pause the video, or while I do it on my end, you can do it on your end and see how it's going to turn out in your notes. Now, when you're doing this, be really, really careful because there are so many letters, it's quite easy to get to fill in the wrong thing if you're not concentrated. So I'm going to stop talking and keep going while you guys fill up your table and your notes. Just want to remind you that because this is a complete dominance scenario, we're not going to work with anything more complicated than complete dominance. The big A represent the dominant. The lowercase will represent the recessive. And when you write out genotypes like these, always put the dominant at the front. Okay, so there we go. I have completed. And hopefully I haven't made any mistakes while I do it. Now let's do genotype together. So once you got that, we have the genotype ratio. So let's just write it out. For the genotype ratio, you'll actually do exactly the same thing as you would have done before. So we will look at the individual and how many of each we get. So the first one, let's just put the homozygous dominant. So we don't know how many AA and BB, to a certain number of, looking at this, big A, big B, little b. And then we also have a combination looking like this. So it's a certain number of big A, big B, and then we have a certain number of this, which is big A, big B, little b. A, big B, little b. And then let's see if we have any other combinations. We could have, we already have this. We could have big A, little b. And do we have this one already? Yep, we do, which is right here. Okay. However, we do not have this one. So let's do that one as well. Big A, little b. And then we have this one already. Yes, we also have this one in our ratio already. We haven't have this one. So little a, little a. So little a, big B. And then little a, big B, little b. And then the rest of them, there's just one more, which is little a, little b, little b. Okay, so there we go. We have written down all the genotypes that we saw in the Punnett square. So when you do a genotype ratio like this, always write down all the different combinations of the genotype that you see. And then you look at the number of those genotype that you see. So the first one right here, big A, big B, looks like this is the only one that is present. So I will use a different color. So we only have one. So let's put a tick right here. And then the second one, big A, big B, little b. We have one right here. And then do we have another one that display that? Yep, this one also shows that. See if you can find any other ones. So at the moment, I don't think I see it. So I'm gonna put a 2. The third one, big A, little a, big B. So this is one of them here. And here is another one. And at the moment, I don't think I see any other ones that looks like that. So I'm also going to put a 2. And then the third one would be this, big A, big B, little b. Now I see this one looking like that. This one also looks like that. This one as well. I don't know why I wrote that, but that should be a B. Yep, there we go. So 1, 2, 3, 4. Okay, now the next one, big A, little b. So I see this one right here, and that's the only one that I see with that particular combination. So that'll be 1. And then the next one, we have this. So big A, big B, little b. And this one's the same. So we have 2. And then this one, we see this. And that's the only one that I see at the moment. And then we have this one, and I see this one and this one. So there are 2 of those. And then there we go, we have 1. So last box ticked and written down. So this is how you would do a genotype ratio. All right. Now let's look at a real life example together. In pea plants, there are actually 2 genes that determines the physical appearance of a pea. And that would be actually what I want you to do now is after you've done that, I want you to write down the gamete allele combination practice thing. So practice coming up with the different combinations of alleles in the parent's gametes. Now, one trick I can teach you is, let's say if I have, let's say if I have a genotype looking like this, A, B, B. My quickest way to do this on pen and paper would be take the first one, that's number 1. This is your first sperm. Take the first one and the second one of the second gene, and that's going to be big A, little b, and that's your second sperm. And the third one is take the second one and the first one of the second gene, and there you have a little a and a big B. And then the last combination would be taking this one and this one. So you have a little a and a little b. So that's your other sperm. Now, if you do it like this, you can make sure that you didn't miss any of the combinations, as long as you see 2 lines at the bottom of each of your allele in the parent's genotype, right? This is my trick to doing it. Make sure I get the step correct, because this is the hardest step to get wrong. So in your little practice of making up the gametes, allele combinations, use this method. Grab some pen and paper and do it. All right, yes, now we can move on to the real life examples, given that I know you've already done your practice. So the real life example, there are 2 genes, and one of them is the color and the other one is shape, all right? So shape and color. Now the 2 alleles for the gene for shape would be round and wrinkled. So if it's round, it kind of looks like this smooth surface. If it's wrinkled, the surface is wrinkled. And then there are also 2 alleles for color. They could be yellow or it could be green. So in this case, the color yellow is dominant over the color green. So this yellow color, this is for green. So Y is for the color. And then the shape is represented by R. So big R, which is a dominant, that is round. And this is wrinkled. All right. So looking at the uppercase and lowercase, you will know which one is dominant and which one's recessive. Now let's say if you have 2 plants and they're heterozygous for both genes and they're trying to produce offsprings, what kind of ratios are you going to see? Now here's your answer. Genotype of the 2 parents, if I tell you they're heterozygous for both genes, that means they will have, oops. And the combinations of alleles in the gametes of these parents, it would look like this. First one would be this. Second one would be YR. Third one, fourth one, like that. So these are all the possible combinations of alleles you can get in the gametes. Now, after you've done that, you'll fill in the gametes in the table. So the gametes will go this way and this way down. And because they're both heterozygous for both genes, both of the parents have this genotype and both of the parents are going to have these combinations in their gametes. And if you fill out the table properly, this is what you'll see, all right? So according to the table, again, you're going to do your genotype ratio. So I'm going to say 1 YY RR 2. These 2 are the same. So Y 2, big Y, big R, little r. And then these 2 are the same. So 2, big Y, little y, big R, big R. to this one and this one. Actually, these two are also the same. So actually, that's 4. And then big Y, little r, there's only 1. And then these two are the same. So 2 of the big Y, little r. And then this one, there's just 1 of them. And these two, and finally the last one. So that's your genotype ratio. Now, when you look at the phenotype ratio, we're looking at physical appearance. So physical appearance wise, it's slightly more complicated. You need to kind of look at the overall phenotype of what this allele is what this genotype is gonna give you. So usually the first one will write the dominant complexion. So this will be yellow and round. Now let's look at all of these and see which ones are yellow and round. So this one is definitely yellow and round. This is yellow and round because it has a big Y and one of the little R's. This is yellow and round. This is also yellow and round. And that's it. So let's count how many of these are yellow and round. So 1 plus 2 is 3, 5, 9. So 9 of them are yellow and round. All right, I'm going to use a different color for the second genotype. This one, let's have green and round. So green means that it needs to have 2 of the little Y's. So this has the little Y, but then it has a dominant round shape. So that's one of them. This one is also green and round. And that's it. There's only 2 of these. So 3 of them is green and round. And then let's use a different color. Let's use pink. This time we want to say yellow and wrinkled. Now this is yellow and wrinkled. This is also yellow and wrinkled, and that's it. So 3 of them are yellow and wrinkled. And then the last one is green and wrinkled. So this one. So 1 right here. And this is how you would do your phenotype. Okay, so finish the practice questions and answers will be uploaded.